the lecture online, so it's all good. Yeah, given that, I'm going to get going, so maybe we can get out of here early. I will point out I'm on my second cup of coffee, and I may lecture quickly. So one way to slow me down is to raise your hand and ask a question. Before I really get rolling too, so I've been doing this lecture. I could just click on autopilot and just talk aimlessly for the next 90 minutes, but I won't. I'll try not to. And I've been doing this lecture for like 20 years. And one thing that students often get confused is I will give you like an alphabet soup of transcription factors and growth factors. Don't memorize that stuff. Who cares? It's the concepts that are important. And if you want to look up what a factor does, you go to gene cards really quickly and you'll learn all about it, if you really want to know. However, you will need to keep clear that distinction between a growth factor and a secreted factor. So, who's going to be kind enough to volunteer what the difference is? There's a pretty big difference. What's the difference between these two different types of factors? What's a growth factor? Come on, somebody. Have you guys not had any coffee yet? Jesus. You're gonna make me point to someone and say, take a stab at it? Macy.
Yes.
I see you looking down at the confusion of this. Right here. It's a growth factor.
Say I would know the exact definition of a growth factor, but it's a factor that's presented that helps with like proliferation and
How does it act? What distinguishes it? Because how it acts is what really distinguishes it as a growth factor.
like the type of signaling you mean it uses?
Yeah. So what does it act at?
I know we talked a lot about target-derived growth factors in our past lectures.
Right, so they'll act at receptors that are either on the cell surface or nuclear hormone receptors, which are in the cytoplasm. So growth factors are spit out of cells. They can act on the same cell that it came out of, so autologous signaling or parallel signaling to neighboring cells, and they diffuse away. So it's a factor that's spit out, secreted, and it acts at a receptor, and the receptor is either at the plasma membrane, or there's obviously some factors too that are like nuclear hormone receptors, and somehow they weasel their way through the plasma membrane to act at these nuclear hormone receptors, like retinoic acid, thyroid hormone, glucocorticoid receptors, nuclear hormone. So try to keep that straight. All right, transcription factor. Very different from a growth factor. You guys are all shy and stuff. So a transcription factor is a factor that has a DNA binding motif. And that motif will bind to something called a DNA motif. And its DNA motifs are super duper duper highly conserved pieces of DNA that are in the promoter region of a gene. What's a promoter? You guys, all right. All genes have promoters, and it's a piece of DNA that regulates its whether or not it's expressed. Transcription factor will bind to motifs that are usually found in the promoter. And the promoter is within about 2,000 base pairs of the start codon. That's called the cis regulatory region, okay? And I will go over a lot of transcription factors and growth factors, try to keep them separate. I will try to highlight what they are and what they're doing. Just keep them separate, but know that a transcription factor regulates transcription. So either shuts it down or activates it. It can be a transcriptional activator or a repressor. Usually it acts within that cis regulatory region, just upstream of the start codon, okay? All right. Okay, so remember, don't memorize because it's going to be a ridiculous alphabet soup of factors. Remember that distinction between a growth factor and a transcription factor, and we'll all be fine. Okay. All right, so this is just a preface. Things to keep in mind as I lecture. Who wants to take a stab? What's competence? What does, in the context of a neural stem cell, what does competence mean? What's it competent to do? All right, this is obviously new terminology, so you guys are all very shy because I'm clearly very intimidating. I've been told that a lot. Anyways, Competence refers to the ability of a neural stem cell to produce particular types of cells. So, a neural stem cell is competent to produce neurons and glia, right? The hemopoietic stem cell is competent to produce blood cells, right? So, different neural stem cells have different flavors, and of course there's ultra-pluripotent stem cells, like in an embryo, that are capable of producing everything, right? And then as development proceeds, their competence gets restricted. So that's something to keep in mind, because neural stem cells go through these phases during development, and it's ordered in time. I'm going to talk about neighboring cells and how they influence each other and signal to each other, either via growth factors or cell contact, and how they influence each other to stay differentiated or undifferentiated. So, anyone want to take a stab at, like, what's an undifferentiated? Cells are differentiated, right? Are they? What's an undifferentiated cell? Well, how would one define that? It's verging on self-explanatory. Anyone want to take a stab?
A cell that hasn't fully committed to its lineage.
That's good, but what actually does the undifferentiated mean? So, I guess contrast with like a fully differentiated cell. What defines a fully differentiated cell?
Characteristics of these ones that are different from the other cells.
Close. How about it has more or less a stable phenotype and an assigned set of functions that it does, right? So like, an astrocyte has an assigned set of functions, right? It's gonna synaptic support, structural support, right? The glia is literally Latin for glue, structural support in the nervous system, and their phenotype is relatively stable. You can tweak it, but it's relatively, so that's a differentiated cell. So an undifferentiated cell, based on what I just told you, is a cell that doesn't have an ascribed function. It's just there. What is it doing? It's not expressing all those genes that make it do useful things in a tissue, right? If you think about a tissue, there's all different cells that have different functions. They are differentiated. So undifferentiated is the opposite. I'm going to talk a lot about the balance of development. Of course, development is quite complicated, and it's a balancing act between giving proper numbers of cells and proper numbers of different types of cells, and assigning cell fate. I'll talk about that, and feeding into that too, like questions just to think about. And this is like, you can expect questions like this on the exam. You know, like what if you induce gliogenesis early? What's that going to do? So glia are always produced toward the tail end of development, but let's say we're going to mess with the system and induce gliogenesis and suppress neurogenesis early when they're normally not made. What might that do? You know, use your imagination. And then finally, because I can't help but talk about stuff that I enjoy, why are there so many neural stem cell factors also expressed by glial cells? And can glia act as stem cells in the mature nervous system? And I'm just going to talk about that a little bit right at the end. So why is total cell number really important? Why, when you're making an organ and maintaining an organ, whether it's a brain or a liver or a kidney or a toenail, why is it important that the size is regulated? And there's a Nobel Prize in this, and nobody knows the answer. So, but why is it important? Who wants to take a stab at the potential of getting a Nobel Prize? Why is organ size so important? Does anyone work in cancer lab? Do tumors care about organ size? Not one damn bit, right? So that's why it's important. If you have cells that don't care about somehow determining that the organ is the right size, you're gonna get a tumor, you're gonna die. So that's why it's really bad. And there's not really a good answer for that. People don't know what regulates organ size, and that's why in-part-human development is fascinating, because clearly, under the normal course of development, all your organs know how to do the proper thing. All right. Development is complicated, and all these little arrows represent numerous different steps, and they're ordered in space and time to take you from an undifferentiated neural stem cell into a highly differentiated neuron that has a highly specialized function, form tens of thousands of synapses, and integrate information and send action potentials to targets in a meaningful way. Likewise, oligodendrocytes and astrocytes are really very highly specialized and differentiated, and these neural stem cells are multipotent. Who wants to break down that word and define it for me? Gee, what a surprise, no one wants to talk to me this morning. Multipotent means that it has the ability to form multiple different cell types, right? So a pluripotent stem cell basically says you can make a whole new animal, all the different cell types. Clearly a neural stem cell, back up, is a multipotent stem cell. They can produce interneurons, many different flavors of interneurons, projection neurons, oligodendrocytes and glia, and this is work that goes way back into the 80s using viral lineage tracing. Before that, people didn't know, oh, there's a precursor that makes only astrocytes and one that only makes oligodendrocytes or this type of neuron. And it turns out these neural stem cells are quite plastic, and they're capable of producing all the different cell types with different competence windows. I'm going to get back to that in subsequent slides. But they're multipotent. So if you tag an individual neural stem cell with a virus that has a reporter, and you follow it, so let's say it's a retrovirus that integrates into the genome, so it's not going to get diluted out, and you can look at all the clones that are made from it. You can find that that stem cell gave rise to a bunch of astrocytes, oligodendrocytes, and different types of neurons. So multipotent. Okay. And here really the take-home message is that there's really a lot of different steps that are ordered in time, and you can perturb this, and the system is amazingly plastic and it compensates. So try to remember that, and I'll show examples as we go through along here. So I did hear that you guys did a little bit of the neural tube and neural induction. I'll go over a little bit of that, and I'll go over a little bit of jargon here too. I already talked about secreted and transcription factors because that's really important because they do very different things, and it's important to keep that straight. Symmetrical versus asymmetrical divisions. So of course, early in development, neural stem cells do nothing but symmetrically divide. They give rise to daughter cells that are equivalent, like two neural stem cells. That's a symmetrical division, right? Asymmetrical obviously implies that one daughter cell goes on to be a neuron or a glia, and the other one stays a stem cell. So there's symmetrical versus asymmetrical, and in the early neural tube, it's nothing but symmetrical divisions. I already talked a little bit about competence and directed potency we've gone over, and then I'm going to go through like this transcriptional code, this alphabet soup. And again, don't remember all the factors, it's just to give you an idea of how these mechanisms come into play. As I mentioned, early development, it's predominantly just neural stem cells doing neural stem cell things, and then eventually neurons are produced, and then the glia are produced. And I'll talk about proglial transcription factors and secreted factors and how clearly if it's a profactor, it tends to support it. There's also inhibitory factors that tend to suppress it, and we'll go over a little bit of that stuff. I don't even know why microglia in here because they're not produced by neural stem cells. They come from mesenchymal or even yolk stem cells. All right. Okay, this is just a quick reminder. I think John went over some of this with you guys. Right, so at first there's a neural plate which is derived from ectoderm. So basically a thin layer, monolayer of cells that's going to go in the form of skin. And you have to get inhibition of certain pathways like BMPs and WNTs that allow these epithelial cells to become neural stem cells. The plate rounds up and forms a tube, and that tube at early stages of development is nothing but neural stem cells that proliferate and make more neural stem cells. This is just a little bit of old school neuroanatomy that persists from a developing embryo into an adult. So if you're ever doing adult neuroanatomy, you'll come across terms like sulcus limitans and roof plate, floor plate, and blah, blah, blah. I don't think you guys get much neural crest, which is too bad. Neural crest are really cool stem cells. So the neural crest actually, The neural crest actually comes from this dorsal aspect of the neural tube as it closes. As it closes, it looks like a wave that forms a crest. And these neural crest cells migrate away from that, and they go all over the body, and they form all the melanocytes in your body, except for the retinal pigment epithelium, that's neural tube. They form stem cells in your hair follicles. They form all sorts of interesting stem cells, neural crest derived. And unfortunately, I don't think we have time to go over it, but they're really cool cells. All right. I would like to advance. All right. This is just a scanning EM. Pretty sure this is a mouse spinal cord. Oh yeah, neural crest form dorsal root ganglia. So for those of you that study dorsal root ganglia, neural crest, here's your dorsal root ganglia cells. These are all neural crest derived. Anyways, so this is a real world view, so to speak, of scanning EM of neural tube. And if you take out a section of that, like I highlighted right here, and then you draw a schematic diagram, basically the wall of the neural tube is nothing but neural stem cells, and the purpose of these guys is just to proliferate, and they undergo this big process called interkinetic nuclear migration, which basically means as they go through different stages of the cell cycle, S phase for DNA synthesis, and then into G2 and M phase, and back into G1, and then decide whether to re-enter the cell cycle into S phase, their cell body translocates to different layers of the neural tube, and this is just called interkinetic nuclear migration. I'm going to talk a little bit about this later because this may actually serve a function. It's not clear that it's anything other than just helping to segregate different developing layers of the neural tube. So all the mitotic figures are always tend to be aligned at this ventricular surface. So where there's a ventricle or the neural tube, the center of the neural tube, that's where these mitotic figures, cells in M phase, are found, and they move. And then as they differentiate, they migrate apically, so toward the peel surfaces. Same information in a slightly different format from a review from Mike Dyer and Rod Bremner. This goes on in the retina as well, where there's not exactly a well-defined neural tube, but of course the retina is out pocketing of the neural tube. Similar things happen, again, interkinetic nuclear migration. And then as the cells differentiate, another thing that keep going over, typically the first cell type made are large projection neurons. And that's just a theme that's consistent, in development. So in the retina, it's ganglion cells. In spinal cord, it's going to be motor neurons. In cortex, it's going to be superficial pyramidal neurons. Or sorry, deep pyramidal neurons, because they get deposited. So I don't even think we probably don't even go over that. But the deeper layers of the cortex, those cells are made first, and then the newer cells climb through them and get deposited on top, and then you get the more superficial layers of the cortex. But it's typically the projection neurons that are made first. Okay. This is just from a review article from Ryuichiro Kageyama. I'm actually going to talk about a few papers from his lab. Really some beautiful work. So again, kind of outlining this time from left to right, symmetrical divisions, asymmetrical division, and then a gliogenic phase. And one thing I'm going to point out here, it kind of shows it as a very discrete switch. What it is actually, it's a probabilistic switch. So you'll get a decrease in probability that the neural stem cells are going to make daughter cells that form a neuron, and an increase in probability that the daughter cells are going to become glial. It's not a discrete and absolute binary switch. But of course the trend is that early in development, neural precursor is doing nothing but proliferating. Then eventually you get asymmetrical divisions where you get the first neurons being produced, which are, remember, typically large projection neurons. Interneurons are made later. And then when you get sufficient numbers of neurons being made, signals are produced that say, tell the progenitors, all right, let's change your competence and let's increase the likelihood of producing glial cells. Same information in a slightly more detailed format. And here, what it's doing is rolling in these neurogenic signals, which are these growth factors that are secreted. and these proneural genes, and these are transcription factors that are cell autonomous. I will point out, we're going to keep life simple. Some of these transcription factors actually get secreted and can do all sorts of stuff too. But 90% of the more than 90% of the function of these transcription factors is to regulate DNA access, expression, transcription. In this figure, what I'm going to point out is that early in development, represented by this little skinny arrow, these progliogenic signals are relatively small, and then you get a big fat arrow where these signals are elevated later in development. And likewise, these proneural genes that say we need to become a neuron are prominent, as are these neurogenic signals, and then later in development, these signals get diminished. And then of course, you also get feedback from these gliogenic signals that say, nope, we don't want these proneural factors. However, 20 years ago when I got started, the field of adult neural stem cells was really super duper popular. It's like, we're going to be able to cure all the diseases of the brain with these neural stem cells and transplant them. And of course, that didn't happen. But there are, in some species, bona fide adult neural stem cells that have plasticity and can genuinely participate in regeneration. All right. So now we're going to switch from that initial phase of just stem cells doing nothing but boring things other than proliferating. Hopefully not too much, because we don't want to get a tumor. To switching to a gliogenic fate, or sorry, a proneural fate. So the key factor involved here is Notch signaling, and the ligand delta is actually embedded in the plasma membrane. It's not. There are secreted Notch ligands, but they basically don't do very much, because it's this physical interaction that causes a stretching of the Notch ligand, and stretching of this Notch ligand allows it to be proteolytically cleaved and liberate an intracellular domain to elicit cell signaling. But basically what happens is early in development, all of these progenitors have this Notch, delta Notch signaling, and keeping themselves undifferentiated, and at one point in time, the one cell gets enough proneural genes and it upregulates the ligand, the delta or jagged or whatever, to keep other neighboring cells as progenitors. And when enough proneural genes accumulate, then you get an inhibition of glial genes, less activation of, proliferation inhibitors like the cyclin-dependent kinase inhibitors, and then these proneural genes. So here's where the alphabet soup starts kicking in. So I will go a little bit more over Notch signaling because it's quite important. All these genes here are neurogenic BHLH factors. So BHLH, this thing automatically branches. Anyways, is basic helix loop helix, and this is a motif, and they bind to promoter regions, and the motif is called an E-box. So it's a consensus region of about 20 base pairs, it's highly conserved, and these proneural genes bind to that and they influence transcription. All of these genes here are neurogenic BHLH factors, and they promote neuronal differentiation. So you get enough of these genes accumulating, and these genes are all suppressed by Notch signaling. You see the Notch inhibiting this, and of course these genes feed back to inhibit these proglial genes and Notch signaling genes, which also happen to be BHLH factors. I'll talk about that in a minute. Okay. So you'll have a neighboring cell with, here's delta-like 1, the ligand, it'll bind to Notch, it'll stretch that receptor, it'll get cleaved by a gamma secretase, and unlike most other cell signaling pathways, there's no signaling cascade to amplify the signal. You get activation of one receptor, and you get one transduction of this Notch intracellular domain, if it escapes inhibitory factors, finding its way into the nucleus and then interacting with other factors. So complexing with this mastermind-like gene and RBPJ. I'm going to talk about RBPJ right at the end because it's kind of interesting. And with this notch signaling, you get activation of these Hess genes. So hairy and ants are split. Gee, what a surprise, it came from a Drosophila strain. The flies were really hairy. The activation of these are pro-progenitor and pro-glial BHLH factors, and they suppress these proneural factors. This is now called ASCL1 neurogenin 2, and these keep cells in an undifferentiated state as a progenitor, and later in development, they promote glial differentiation, particularly astrocytes. So we'll revisit this notch signaling in astrocytes and glia later. But this is basically what keeps these progenitors as progenitors, and eventually this signaling pathway gets overrun by these proneural BHLH factors, and you get a neuron. Similar information in slightly different format here in the retina. All these PAC6 positive retinal progenitor cells, notch delta, tug of war, and then eventually you have one escaping the cell cycle with sufficient levels of proneural BHLH, turning into a projection neuron, in this case, a ganglion cell. Right, so basically the take-home message is for some magical way, either there's a buildup of notch inhibitor, such as NUM, a cell gets less notch and then enough proneural BHLH to turn it into a neuron, or increase the chances of it becoming a neuron. So this work is really cool, and this is again from a Ryuichiro Kageyama, but the original research paper, they had destabilized reporters, so they could look in real time and did live cell imaging on these cells. So these little waves here represent like the level of fluorescence, and the fluorescence, these were fluorophores conjugated to a neurogenic BHLH, neurogenin 2, or HES1, and what you find here is that there's opposing waves because they mutually inhibit each other. So the more HES1 you get, the less neurogenin you get, and vice versa, because they mutually inhibit each other. And then at some point in time, in these neural precursors, so again, time from left to right, you get enough level of the neurogenin 2, it says, we're done with the HES1, and you're going to become a neuron. So this is again highlighting, and that's how that experiment was done, which I think is really damn cool. In real time with these destabilized fluorophores linked to these, back here, these neurogenic and gliogenic or progenitor BHLH factors, showing this mutual opposition within individual cells, how these levels fluctuate completely asynchronously, and eventually you get one of them winning to become a neuron. Any questions? Yeah.
Is the gray cell like fully differentiated, or are they both like developing through this process?
So this is, I think the gray cell here is the one that's going to be just a neighboring cell. It's probably a progenitor, right? And this green cell, presumably, is where you've got, you know, this HES, this notch proneural factor, tug of war, HES1 fighting with a proneural BHLH. Doesn't necessarily have to be neurogenin 2 in one way or another. So this is just a neighboring cell, the gray. Okay. Another thing that might factor into this, you remember I talked about this interkinetic nuclear migration. One possible function is that there's a gradient of receptor and ligand. And at this apical surface where all these cells are in M phase, you might receive a little bit more notch. So one can imagine if these receptors are concentrated primarily on the cell body, which is moving, you dip further into this pool of ligand, which isn't diffusing, right? Because it's in the cell membrane. So you dip further into this pool of ligand, you're more likely to stay a progenitor. However, if you migrate less far into that gradient of ligand, you're more likely to become a neuron. So that's one possible reason for undergoing this interkinetic nuclear migration. Yeah, it's all spelled out there. Right, auto advance is fine. I'll deal with it. All right. So I threw in this factor, or this slide here, just to also point out, you know, again, you have this tug of war between these proglial factors and these proneural factors. For now, just ignore id factors. These are very proglial factors, but let's just focus in on HES1, HES5, fighting with these proneural factors. And again, pro-neural secreted factors, now we're talking about factors that are spit out of cells and act at receptors at the cell surface and activate second messenger cascades within the cell. And these can also promote these pro-neural factors, and this extracellular milieu, this environment changes, and of course, at the right time, lots of FGF and lots of neurotrophins helps you make these progenitor cells and the progeny form neurons, and then later you get all these proglial factors. So epidermal growth factor, ciliary-derived neurotrophic factor, bone morphogenetic protein, and these second messenger pathways such as JAK-STAT and SMAD signaling, which tend to be proglial, telling these progenitors, all right, maybe we've got enough neurons, let's start increasing the probability of making glia. Does that make sense? So if you were an omnipotent being designing a system, what do you think would be a good source for these proglial factors? If you were to develop a feedback system. And again, keeping them in mind that you want to get proper numbers of neurons, because even too many neurons is a bad thing. I'll share an anecdote in a minute. Oh, back here. Well, what would be a good cellular source for these proglial factors? Yeah.
I'm just gonna guess neurons.
Yes. 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 Very good. Right. So you get enough neurons, and they produce. Let's say each neuron produces just a little smidge of growth factor, like EGF or CNTF. You get enough of them, and then it says, "All right, got enough neurons. Let's do something else." Perfect. And that's indeed the case. And I'll go over a specific example of that shortly. I will point out, I should probably include this. It was a science paper from Chris Walsh where he deregulated proliferation to the point that it was a mouse knock-in animal, and he messed with Wnt signaling, and the progenitors proliferated so much that the mice got cortical folds. So no longer a smooth cortex in mice, they got cortical folds, like looked a little reminiscent of higher vertebrates that we had a folded cortex to accommodate more neurons. And so this is probably early 2000s, and the media lost it. Scientists at Caltech is making mice with human brains. And everyone's freaking out. So, who wants to take a guess? Were these mice smarter? Raise your hand. They were dumber. If that's even possible. Yeah. So they had way more neurons, but man, they were even dumber. Anyways, so having more neurons is not a good thing, and that's why development is important. Don't want too many neurons. Clearly don't want too few neurons. Right. Fun little anecdote. All right. So of course, there's more than one type of neuron, as it turns out. And then what determines different types of neurons? And I'm going to show you a lot of detail here. Don't memorize the detail. It's basically a combination of secreted factors, growth factors, and a transcription factor code. So this was really worked out in the spinal cord absolutely beautifully by Tom Jessel, who we apparently don't talk about anymore because he was bad. He was Howard Hughes funded, and at one point in time he had 40 postdocs in his lab. And all the different antibodies, like from the hybridoma banks, they all came from his lab. Just unbelievable. Anyways, I think this is actually a chick spinal cord. You can stuff a chicken mouse, and then this is just immunolabeling. And of course, all these factors here with an X are a homeobox factor. And a homeobox is a protein motif that binds to a particular DNA motif. So these are all transcription factors. If you see an X, so paired like homeobox 6, Iroquois, X, that's a homeobox gene. Homeobox genes are transcription factors. OLIG2 is a BHLH factor that tends to persist. I should point out too, these neurogenic BHLH factors tend to be transient. So they'll go up you'll get a neuron and then they'll go away, and then these homeodomain factors take over to stabilize the phenotype. So, clearly what you can see here, that there's gradients in patterns of expression, and from this dorsal ventral axis in the neural tube, here's your motor neurons. And motor neurons, you want to get a motor neuron? Well, you need OLIG2 and some NKX 6.1 and maybe a little bit of PAX6. Boop, motor neuron. And there's your transcriptional code for these different cell types. Does that make sense? OLIG2 does persist, but it's not really a classical neurogenic VHLH factor. It is a VHLH, but ASCL1, Neurogen, and NeuroD, they all tend to be transient. All right. Same information, slightly different detail here from Francois Guillemot, looking at the different types of motor neurons down here and different types of neurons in the spinal cord that I'm completely unfamiliar with. And again, to get the different types, it's combinations. So it's a combination of a homeodomain factor, PAX6, NKX2.2, plus a VHLH factor. MASH1 is now ASCL1, neurogens, even some Hess factors. Of course, these Hess factors are going to tend to give you glia, and this is how you get different types of cells along this dorsal ventral axis in the developing neural tube. Dorsal neural tube, same thing. Again, gradients of transcription factors give you all these different types of, again, atonal 1, neurogenin 2, ASCL1, these are all neurogenic VHLH factors, which can also inhibit each other, and islet1, brain3, these are, and LMX, these are actually all homeodomain factors. And here you can get the different subsets of distinct neurons in the dorsal aspects of the differentiating neural tube. Yeah, it's always more complicated. So this is just to show how it's been worked out in detail. And look at this bloody alphabet soup. And again, all these are VHLH, all these are homeodomain. Look at them all. Why does it have to be so complicated? Jeez. This is just to point out that it's complicated. Don't learn any of this crap. Not worth it. All right. So another thing I'm going to point out again, just to drive home, retina works the same way. And here, each curve represents the probability that at that point in time, so if you, let's say at this point in time, this is going to be, let's say embryonic day 14 in a mouse, a little bit earlier, how about 7, later. You're going to get a good probability of getting a ganglion cell, horizontal cell, amacrine cell, maybe a cone photoreceptor, and then later in development, you're more likely to get a bipolar cell, a rod cell, and then finally the mutar glia. So this is probabilistic, and again, this reflects this notion of these competent states in the retina, and of course that holds true all over the place. So here's like specific examples. This is for a chick retina. And what's pretty cool, whether you're a zebrafish or a chicken or a lizard or a mouse or a human, this pattern of differentiation is consistent. These cell types are consistently formed in these patterns. Again, it's probabilistic. And you end with the probability that your terminal differentiation, when these progenitors say, we're not needed anymore, come back here. We're going to make 2 mutar glia, which are the equivalent of astrocytes, but just in the retina. Again, same thing this is the mouse retina. Of course, the mouse retina is rod photoreceptor dominated, so you get a much larger probability peak for generating rods. Same information, slightly different format. And again, what's interesting too is these progenitors, you'll have a large gamish of the alphabet soup of transcription factors, and then as differentiation proceeds, you get much more refined expression. This is absolutely terrible resolution microscopy and imaging, but the take-home message is that all of these transcription factors, REX, PAX6, 6.3, and this is partial too, there's also SOX2, SOX9, LHX2, blah, 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 blah. Go on for days. Eventually, they get segregated into the mature neurons to give you distinct patterns and transcriptional codes to fake all the different type of neurons in the retina, or spinal cord, or cortex, or wherever. And again, just to drive home that message, it's actually that combination of BHLH factor, MAF5, which is now called atonal 7. Oh, it's so confusing. So that's another reason. Don't remember the alphabet soup because it changes. All the progenitors have the big list, and then these factors get segregated with the help of a BHLH factor, such as atonal 3 or ASCL1, whatever. Or the Hess factors to get you a glial cell, the Mueller glia. And I'm gonna, yes?
Sorry, would you at least want us to know like an example of the BHLH factor?
Nah.
Okay.
And right, if you think it's a BHLH factor, go on your phone or on your computer, go on Gene Cards, and you'll find out very quickly what it is.
Okay.
Just know that BHLH factor, that it can be either very pro-glial or pro-neural, and they're usually transiently expressed. And then again, they usually need to be combined with a homeodynamin factor to give you a particular flavor of neural.
Okay.
Okay. So this next example is specifically to make your brains hurt a little bit and to demonstrate this notion of competence. So you knock out MAF5, now known as atonal 7, you lose all the ganglion cells. Bye-bye. Nadine Brown and Tom, Joe Brzezinski's on this paper too. They're now at UC Davis. And anyways, really cool work. You knock it out, you don't get a single ganglion cell. So clearly MAF5 is required for that competence in progenitors, which is really interesting. So given that, do you think that MAF5 commits? So if a progenitor is MAF5, this is a proneural BHLH factor, you knock it out, you don't get a single ganglion cell, or almost none, almost all gone. So if a progenitor expresses MAF5, who thinks that you're absolutely going to become a ganglion cell? They're going to be, that's the key. That is the magic thing. You knock it out, you don't get a single, if you express it, if a progenitor is MAF5 positive, will it be a ganglion cell? Yes. Stop it. How to advance. Who thinks that? Yeah, you know I'm setting you up. Of course not. It conveys competence. So you have to have it to make a ganglion cell, but if you're MAF5 positive as a progenitor, it doesn't guarantee that you will be a ganglion cell. It conveys that competence window. So, all right, now I can see. More nice work from Joe and Tom. So here they used a bacteria artificial chromosome. So this is 30 to 40,000 base pairs. It's a big damn chunk of regulatory DNA. So it's not just that cis regulatory region that's about 2,000 base pairs in front of the start codon. This is a super huge, large regulatory region of MAF5, 30 to 40,000 base pairs. So it's a MAF5 promoter driving Cre recombinase to a human alkaline phosphatase and a stop codon. So any cell that is going to be MAF5 positive is going to get this reporter. So this is an old, it's not, nowadays if they made it, it would have been, of course, GFP, TG tomato, whatever, but this is an enzyme that you can react to get a chromophore deposition. Okay, do you understand it? And this is what they found, which I think is really cool. Different cell types. So if they look at the clones, right? So once you're MAF5 positive, you're going to have this alkaline phosphatase being expressed forever. These cells are doomed to express human alkaline phosphatase forever. So in a normal retina, this is the relative distribution of cell types. Of course, there's a boatload of rod photoreceptors because mice eyes are designed to see little bits of shit in a deep dark tunnel. However, if you get rid of MAF5, you'll know you get virtually no Mueller glia, and you get compensation. So you still, the total number of cells in the retina is roughly the same, which is pretty cool because we don't want to get tumors, and these programs are pretty hardwired in terms of total cell numbers. You lose the ganglion cells, you lose kind of competence, neural competence in general, but you get compensation. Fewer rods, which is a later born cell type, and more earlier or shifts in different types of cells. However, the MAF5 lineage in the knockout, which is really interesting here, is if you're MAF5 positive, doesn't mean you're gonna be a ganglion cell. So you don't get ganglion cells, but this MAF5 lineage that has this ALK phos in, with this knock-in, they form all the different cell types, even a few mutaglia with mutaglia. So just because you're MAF5 positive does not guarantee that you're going to make a ganglion cell. It just means that you have the potential to form it, and you need that potential or you don't get any cells. And this is a general principle in development for competence. Does that make sense? Okay. Right, so here they're combining this MAF5 reporter with the knockout, and this is what they end up with. So you can still, this reporter can still be active even though you don't have MAF5. Does that make sense? And when they do that, this is what they end up with. That, yes?
I'm still a bit confused what you mean by MAF5 lineage wild type and knockout.
Sorry, say that again?
Is it both a knockout?
So the wild type means it expresses MAF5 protein. The BHLH is there, wild type. No MAF5 here, but you can still trace the lineage with this construct. So MAF5 protein is gone, but the promoter is there, and instead of the BHLH MAF5, you get the reporter, ALK phos. And when you get that, again, no ganglion cells, but it says that these cells still have the program to make, to activate the MAF5 promoter, and that promoter ends up in all these different cell types, which are again, it's reminiscent of this wild type without this MAF5 lineage, right? So this is with the protein and the reporter. Look where all these cells become, which is pretty cool. Saying that MAF5 isn't the end-all be-all determinant of making ganglion cells, which are just right here, right? 11%. 11% total are ganglion cells. It means that if it is there, you have an 11% chance of becoming a ganglion cell, or you'll have it expressed, and for some reason, you'll become a rod photoreceptor, or a displaced amacrine cell, or an ion alpha, come back here, amacrine cell. Does that make sense? Yeah.
Sorry, I don't know, am I tripping, but why is the MAF5 lineage wild type so different from the regular wild type? That's literally gonna be my next question.
Okay. Right, so not all progenitors are MAP5 positive, only some of them are, right? So this is the total lineage. This is the end product if you summarize all the different cell types in the retina. But if you are a progenitor, a subset of progenitor that is MAP5 positive, based on this construct, this MAF5 bacterial artificial chromosome promoter, big chunk of promoter, right? And the protein, the BHLH is still there. It's not going to match the same because not all progenitors are MAF5 positive. But if you are MAF5 positive, here's the probability of your fate. Only 11% of the MAF5 positive progenitors form ganglion cells. MAF5 protein positive, but if you lose that, that goes to 0. So the progenitors, that competence is 11% in this case. So the whole point here is just to say, if you're MAF5 positive, you are competent to form ganglion cells. Without it, forget it. There is 0 chance of you forming a ganglion cell. Does that make sense?
Yeah.
Okay.
Like, isn't this a weird way of displaying that, since that 0.6% in the total retinal wild type is not comparable to that 11% in the lineage wild type?
Yeah, but don't forget, this is all the retina cells, and then unfortunately, I shouldn't say unfortunately, you know, a mouse retina is overwhelmingly dominated by rod photoreceptors, 78% read, right? And then compared to the total number, ganglion cells are only 0.6%, right? And then that's just the average of the total, but of course with MAF5, like the MAF5 lineage, your probability increases. Okay. All right. So that's competence as best as I can explain it. And I think this is a really nice example of it. I'm going way too slow. That's unbelievable. All right. I'm going to go over this stuff. More work from Ryuichiro Kagiyama. This basically says if you lose different BHLH factors, you get shifts in cell types that are made in the retina. Worked out very nicely. So if you lose both atonal 3 and neuro D, you don't get amacrine cells, and you get an overproduction of ganglion cells. So it's the loss of one cell type and compensation by another cell type. Balancing act, very easy. Conversely, if they use viruses to overexpress PAC6 and atonal 3 or PAC6 and NeuroD, same thing. You get if you get forced expression of PAC6 and NeuroD, you get more Amerkin cells. If you get forced expression of PAC6 and MAT3, more horizontal and Amerkin cells, and you get suppressed differentiation of bipolar cells, rod photoreceptors, and mutaglia. Again, balancing act, shifting cell types around. That's the only point here. And just going back to some of the original literature. But that's just the point. It's a balancing act in terms of more of the same thing. Let me go through that. So I gave you an example of a rat and said, oh, it's an Amerkin cell. Lo and behold, 120 years ago, Ramon Santiago Cajal, there's like, he characterized maybe 30 or 40 different types of Amerkin cells. Nowadays, if you do something, what's it called? Single cell RNA-seq? I don't know, something like that. I'm joking around because I do it all the time. There's probably 50 or 60 transcriptionally distinct types of Amerkin cells, 16 to 18 different types of bipolar cells, 20 or 30 different types of ganglion cells. So even within the subsets of neurons, you get further differentiation. And these cells are distinct based on their morphology, their transcriptional profile, and their function. So it's much more complicated. And aren't you guys lucky that we don't have enough time to go into that today? Okay, here's a great example. So photoreceptors, right? I said, photoreceptor, CRX and NeuroD. Well, photoreceptor, nope, not quite. There's your CRX, there's your NeuroD. How about a little bit of MASH1, some OTX2? I don't even know what those factors are. How about some nuclear hormone receptors too, like RXR, gamma, ROR, beta, thyroid hormone receptor. Oh, look at all that feeding back in and making the different types of photoreceptors. Oh my God, it's so complicated. Anyways, that's the only point there. It's much more complicated than the information that I've given you. And of course, laid on top of that is all these secreted factors which have differential effects on the different cell types. That's the only take-home message there. One thing to remember here too, and I do hope you remember this for all sorts of different stuff. All these factors have different functions, and I'm going to go briefly over this one called sonic hedgehog. So sonic hedgehog is actually produced ventrally by mesenchymal tissue, and then it induces expression in the floor plate. So this patterns the entire ventral aspect of the neural tube. And during development, sonic hedgehog has, in the nervous system, 20, 30 different functions, which is kind of similar to a lot of transcription functors. I'll come back here. Have these pleiotropic functions. Pleiotropic just means more than one function. So, and I'm going to go over that really quickly for sonic hedgehog, but if you've heard about sonic hedgehog, again, this is a fly mutant where the fly mutants looked like a little hedgehog, so it's called hedgehog. And then the mammalian ortholog is called sonic hedgehog by someone who played a lot of video games in the late 80s in Tom Justice Lab, who we don't talk about. All right. Quick side note. So for patterning that John Overnick went over, what do you think happens if you get rid of one of these dorsal patterning factors, like let's say BMP4 or WNT4? You knock it out from the roof plate or from some of the cells in the overlying ectoderm. What do you think? So these are opposing gradients. Who wants to take a guess what happens? Imagine this like being an exam question. What do you think would happen?
If we lost both directions or just one?
Now you're getting rid of the factors just from this dorsal aspect. What do you think might happen? In terms of cell fate, like all these different types of spinal cord neurons and glia that have distinct domains from ventral to dorsal. What do you think would happen?
Maybe just cell soup.
No.
I was gonna say that they congregate on the floor plate.
They congregate.
Because they root in the floor plate and don't migrate up towards the floor plate.
No.
They would migrate further because those inhibit them, right? so they would migrate more dorsally.
Not necessarily migrate, but their fates get shifted. So not necessarily migrate, but that's really close. So all the domain of the P0 and the P1 and the motor neurons, they all get expanded dorsally, right? And that's like just an example of these secreted factors that establish diffusion domains influencing cell fate, specification, and differentiation, right? So if you get rid of one of these ingredients, all of these, you're going to end up with more of these ventral domain neurons. Their domain's going to expand dorsally, and you're going to get fewer dorsal domain neurons, all their different names and stuff, which I don't know because I don't study the spinal cord. Okay? All right, little side note. All right, pleiotropic, we're going to go through this pretty fast. Sonic hedgehog, pretty cool. So sonic hedgehog is actually a very lipophilic secreted factor, so it doesn't diffuse that far. It's produced by mesenchyme, and then it's induced in this ventral floor pate. And this is just an example of all the different functions that it can have. Interesting side note. There's a drug called cyclopamine. So cyclo-1, I, back here. And this was a drug, and it's produced by western hellebores that's eaten by sheep, and if you're pregnant and a sheep and you eat it, your offspring are born cyclopic and dead. So if it's eaten just at the right time, because the sonic hedgehog forces this, separate the eye field is just one eye to begin with in the ventral diencephalon. And the sonic hedgehog causes the separation of the eye field, so you get two eyes. And if you block sonic hedgehog at just the right time, you end up with cyclopic babies. Anyways, so cyclopamine, which blocks sonic hedgehog signaling, gives you cyclopic babies in sheep and cows. So there's one function for sonic hedgehog. Fun little anecdote, helps you remember it. It also is expressed by ganglion cells. Someone brilliant said it produced by neurons. This is sonic hedgehog. You get enough sonic hedgehog feeding up, and then come back here. The cell fate changes, and it says stop making ganglion cells, for example. And that's the whole point of these. So if you interfere with sonic hedgehog signaling, you get more ganglion cells, even though it's being produced by ganglion cells, because it feeds back onto the progenitors and says stop making ganglion cells. Also promotes rod differentiation and survival. It promotes lamination in the retina, influences pathfinding in the optic chiasm, promotes survival and differentiation of the pigment epithelium that lies behind the retina. The point here is don't think that a transcription factor or a secreted factor has just one function in development. They're reused. That's the only point here. Okay, gliogenesis. How much time do I have? Just goes till
9:30.
till 9:50. Oh, we have a little bit of time. Cool. Okay. Gliogenesis. You've seen this before. So we're done making neurons, and we're starting to get the neurons producing these magic factors and say, all right, how about a little bit more of these id factors or NFI factors, or a little bit more HES1 and HES5, and let's start making glia. Same information, slightly different format. Again, here I just mentioned NFI. I forgot what it stands for. I should know. I publish papers on this shit. These NFI factors aren't transcription factors, as are id factors, but they just influence transcription factors. And when they build up, so NFI A, B, X, whole alphabet soup of them, redundant factors, which is really interesting. To get an effect, you literally have to knock out 3 or 4 of these factors to inhibit gliogenesis. So it's a robust program that maintains the knock out one. It just doesn't matter. You have to get rid of 3 of them. So kind of cool. Signaling factors come in here, of course, Notch, which we talked about, JAK-STAT signaling, CNTF, whatever, helps to get you astrocytes. Again, take note, late development, you tend to get glia. Cellular sources, nobody really knows, but that's how it works. All right. There are exceptions. So remember too, again, you have these progenitors changing their competence from neurocompetence toward an increased probability of producing glia. However, there are a few exceptions. One of them are these oligodendrocyte precursors in the developing neural tube, and there's also glial-committed progenitors in the developing optic nerve. So there's no neurons in the optic nerve, of course, it's all just axons and glia. So this is the exception to the rule during development. Of course, you also need olig-2 for motor neurons here, but you also get discrete oligodendrocyte progenitors. And here what happens is early in development, these neural stem cells tend to express neurogenin 2 and olig-2. It's not a switch point, it's not binary. There's a shift, and then all of a sudden, neurogenin 2 goes away, and you get, oh look, there's an X in here. So this is a homeobox factor. You get NKX 2.2 plus olig-2, and then you start to get oligodendrocyte precursor cells that are producing predominantly or only oligodendrocytes. Does that make sense? Yeah, I think that's pretty straightforward. Okay. Astrocytes, kind of similar mechanisms, different factors involved. These are growth factors, neurotrophin 3, platelet-derived growth factor, a cell cycle inhibitor to kick these out of the cell cycle, and these astrocytes feed back onto these progenitors to get you differentiated oligodendrocytes in development. So again, astrocytes tend to be formed before oligodendrocytes a little bit later, but of course there's also significant overlap. All right, I already went over this. Same information, different slide, different format. Okay, gliogenesis in the retina. All right, I'm gonna go through this. Right, so clearly a retinal stem cell is very different from a mature glial cell. Interestingly, and this is a short list of the alphabet soup of transcription factors. PAX6, CHEX10, LHX2, oh man, the list goes on. So all these retinal stem cell factors are also present in mature munoglia. How similar are actually glial cells? This is an old study. We've got much better data. I should really update this, but Connie Sapko. Oh, Connie Sapko was actually the one who did the viral lineage tracing. She's like getting awards for that and stuff too, because she's going to be soon retiring. She was the one in the late 80s who did the virus lineage tracing to show the multipotent nature of stem cells. Anyways, in the old days, this is from microarrays, not single cell stuff. They would grab individual cells, harvest the RNA, reverse transcribe it, amplify, and splash the contents onto one microarray. That's pretty cool. And basically what they found is that munoglia, there's about a 70% overlap of their transcriptome with retinal progenitors, which is pretty amazing, because if you compare a retinal neuron to a progenitor, it's about 10 or 12%. So, and let's not be confused or either. These munoglia are bona fide glial cells, structural support, metabolic support, ion buffering support, pH support, all that stuff. Can they become progenitors? Yes, they can. Work that I did a long time ago. And as it turns out, so the retina has been known to regenerate in fish for about 70 years, older than I am, not by much though. And it wasn't only till about 20 years ago, 23 years ago, let's say, that we actually identified, it was completely by chance, better to be lucky than good, in a chick retina, that these munoglia can de-differentiate and become progenitors. And as it turns out, the same thing holds true for a fish retina. And in the fish retina, the munoglia are indeed fully differentiated. You can damage the retina or tweak the munoglia, and they do an absolute fantastic job of giving you functional neurons. Chick, they do it not as well, and the neurogenic capacity is greatly reduced, and mice and mammals suck. All right, but yeah, high throughput, low throughput screen, NFI factors. I already talked about this. If you knock out NFI A, B, and X, I think triple knockout, you can actually turn these munoglia into neurons. And their transcriptional profile is quite similar to interretinal neurons. Don't know if they're functional, but we performed RNA velocity and stuff, and yeah, it looks like they're going in the right direction, which is pretty cool. Keep in mind too, some of you might be aware, is anyone in a lab that looks at like hippocampal stem cells or or subventricular zone stem cells. There's a few labs around that do that. Anyone? No? Okay. So there's cells that line the subventricular zone, so as it suggests, just below the lateral ventricles. Oh, come back here. And these cells look astrocyte-like, and under normal conditions, they're pretty good at producing olfactory neurons. And when they were first identified, just over 20 years ago, people thought, wow, we're going to be able to cure neurological disorders. But it's really hard to get them to stop making olfactory neurons, at least in the mouse. Hippocampus too. They seem to be, again, tons of work from Fred Gage's lab, astrocyte-like cells that appear to be functional, fate-restricted neural stem cells in an adult mouse nervous system.
More recently, there's actually been a number of
COVID. That's why it's auto-advancing, because this is used from COVID, and I've got it. That was my narration. Is my voice really that nasal? Anyways. Oh, I don't want to hear that. All right, so basically if you knock out Notch, you can turn mature astrocytes into neurons. This was a science paper from Jonas Friesen's lab, 2014, and it was recently followed up by my friends Seth and Ton, and you can also do this in Muniglia if you selectively knock out RBPJ, or Notch 1 or Notch 2, you can turn the Muniglia into progenitors in an adult mouse retina. So anyways, I think that's pretty cool. Again, driving home this point that these late stage progenitors that are making glia, the glia retain some character of stem cells, so much so that in a fish retina, in fish nervous system, you get functional restoration of neurons, but you have to tweak them much more in mammals, but it's still possible. And you can record from some of these cells, depending on what you do to them, in an intact retina, and they even respond to light. So, all right. Any questions? This is all in the PDF. I went through that last part pretty fast because I went through the first part fairly slow, so, and the caffeine is kicking in. Any questions? Okay.
Thank you.
Yep.